Welcome, this is a microbiology video explaining why after a gram stain procedure, why gram-positive cells stain purple and gram-negative cells stain pink. And it has to do with their structural differences. Okay, so that thick peptidoglycan layer that's characteristic to gram-positive cells right here, that holds on to the purple stain, okay? And the thin peptidoglycan layer of the gram-negative cells, this tiny little brown sliver, is not enough peptidoglycan to hold on to the purple, okay? Plus, they have this wimpy outer membrane that will not withstand the decolorizer step, okay? So, let me explain. You'll get to do this in lab. There are 4 reagents, 1, 2, 3, 4, and you'll use them in this order to stain both gram-positive and gram-negative cells, okay? That's why it's a differential stain, because you are going to stain all cells with the same 4 steps, but at the end of it, if it has a thick peptidoglycan layer, it's going to stain purple, because it's gram-positive. If it has thin peptidoglycan layer and an outer membrane, it's going to stain pink because it's gram-negative. Okay, that's why it's called differential stain, because it can differentiate between 2 types of bacteria. Okay, so there's the primary stain, the mordant, the decolorizer, which finally lets you see differences between the 2, and then the counter stain, which stains a different color, a color that counters the other color, or the primary stain. Okay, so let's get into the details. Here on your left, you have a gram-positive cell. It has a very thick layer of peptidoglycan. Look how thick compared to on the right, the gram-negative cell wall. Look at this tiny little puny 1 layer of peptidoglycan, and they have this wimpy little outer membrane that's made of phospholipids. Okay. So the first step is the primary stain. This is crystal violet. It's a dark purple solution that is also alkaline or basic, meaning it has a cationic charge. Bacterial cell membranes always all have a negative charge. So what happens with a positive and a negative charge? They attract each other. So the crystal violet, you're going to flood your slides with crystal violet, and the crystal violet's going to get in there, attracted to this negative charge, and just fill in all these spaces of this peptidoglycan of the gram-positive. A similar thing will happen over here with the gram-negative cell, okay? If you were to, now you wouldn't do this because it's weird, because you're not done yet, but if you were to look at the cells at this point under the microscope, they would both be purple. There's no differences yet. Okay, the next step is the mordant. Another way of, I want you to know the word mordant, like this is the scientific term, but I say fixative-like because if you've ever heard the word fixative, it's a similar meaning in that it fixes the ink or the dye or makes it stick so that it won't be washed away easily. Okay, so that's what the Gram's iodine solution does. It's a light brownish orange, okay, and so it's not going to change the color, but what it does, it is still a basic dye, so it's going to be attracted to the cell envelope, and it's going to get into these spaces, and what it actually does is it makes the crystals of the crystal violet larger. That's what it does. Okay, so that's why I tried to depict here. Okay, it's like filling out the spaces in between the peptidoglycan. It's making those crystal violet crystals really big. Okay, and it's doing a similar thing to the gram-negative cell. Now, because it's such a light brown and the dark purple of the crystal violet is so dark, if you were to look at these under the microscope, you wouldn't. But if you were to, they would both look purple still. All right, so that's the second step. It's the fixative step. Step 3. The decolorizer, and it's 95 ethyl alcohol or ethanol, same difference, okay? this decolorizes one of the cells, and I'll give you a hint, it's not the gram-positive cell, okay? Because you have such a thick layer of peptidoglycan, when you douse it in the alcohol, only a little bit of the crystal violet leaves, okay? The rest of it stays. As opposed to the gram-negative cell over here on the right, look what happens. Okay, so you douse it in alcohol. Guess what? This outer membrane starts to dissolve. Oh my goodness. And so there goes all that crystal violet that was there before. Okay, and now you don't see any purple because it's also going to get in here and wash away from this tiny, tiny thin layer of peptidoglycan. It's going to wash away the crystal violet from there too. Okay, so the decolorizer step differentiates gram-positive from gram-negative, okay, because it decolorizes gram-negative cells. Whereas gram-positive cells, if you were to look at it, you wouldn't look under the microscope at this point. You're not done yet. But if you did, gram-positive cells would still be purple, but gram-negative cells would be clear or transparent. Okay? No crystal violet here. Oh no. Okay? But since these are really hard to see, transparent cells are really hard to see, you need to increase contrast, right? You need a stain. You're going to do a counter stain. And it has to be a different color than purple so you can see it, so you can tell them apart. Okay? And so it's red. Safran in solution is red. It's cationic. It's going to behave similarly in that it's going to be attracted and pulled and saturated into these layers of peptidoglycan on the gram-positive cell. But it's a light red, so it's not going to change the color much. Okay? As opposed to the gram-negative cell over here on the right. Look, it's totally clear. It has like barely any outer membrane left and just one little thin layer of peptidoglycan that has no stain on it. So you're going to saturate it with red, a light reddish color, and red plus white, because the background's white, equals pink. Okay? So that's why it's not red, is because it's a light red and you're shining white through it. So it's red plus white equals pink. So that is why you got pink cells for gram-negative and why you still have purple for gram-positive. Alrighty? Because the red doesn't, it's there from the safranin, but it doesn't overpower that really, really dark crystal violet color. Alrighty?